Hello everyone. Today I give a presentation on determining biomolecular signatures and pathways of amyotrophic lateral sclerosis using bioinformatics and system biology techniques. The authors of this research project are Shaila Haq and myself, Tasnim Hosen Tanha. We both are from a department, Biochemistry and Biotechnology, Faculty of Biosciences, University of Borishal, Bangladesh. So the first topic, introduction. The question is, what is amyotrophic lateral sclerosis or ALS? The answer is ALS is a neurodegenerative disease that is predominantly affecting the motor neurons and is incurable. It ultimately leads to progressive paralysis and death within 3 to 5 years of the disease onset. The clinical presentation of ALS is typically characterized by focal muscle weakness and wasting that is adult onset and has a tendency to propagate as the disease develops. Most frequently, the limb muscles are the source of the weakness, with distal muscles being affected more frequently than proximal muscles. In approximately 25 to 30% of the case, the disease manifests as bulbar onset, which is characterized by dysarthria, dysphagia, dysphonia, or in uncommon cases, masseter weakness. Now comes the study part. In this study, we analyzed or used GeoTour tool and the Lima-R package to analyze gene expression data from tissue samples to identify differentially expressed genes with log FC greater than or equal 1 and also log FC less than or equal -1. In the both cases, we always kept p-value less than 0.05. Potential biomarker signatures at transcriptional and translational levels were identified in the analysis, which also predicted drugs for ALS treatment. The focus was on early detection and therapeutic intervention. So the crucial part or step is methods or methodology. Our target disease is amyotrophic lateral sclerosis, I said before, and the analysis, we use a server called NCBI and a tool GEO, Gene Expression Omnibus, where we select a specific RNA sequence data, which is GSE 201407, and we analyzed it through GeoTour tool, and then we find out most important part or information, that is differentially expressed genes. Differentially expressed genes mainly formed by two types of genes, that is upregulated genes and downregulated genes. After finding DEGs, then we analyzed it through gene ontologies and pathway enrichment analysis. Also, we analyzed protein-protein interaction and then transcription factor analysis. And the last but not the least, drug prediction. So now comes the another important part, result. Here we see the data retrieval. In the data retrieval portion, we see two types of picture. First is a box plot, which comes from our target RNA sequence data, and we see a volcano plot. In the volcano plot, we see a blue color portion, which is down-regulated gene, and red color portion, which is up-regulated portion. And then we see gene ontology and pathway enrichment of our target differentially expressed gene or DEGs. This information find out from a server called Enricher. So geoterms. In geoterms, we see three types of biological process, like biological process, cellular component, and molecular function. In the biological process, we clearly see that negative regulation of monocyte chemotaxis is as if top rank, and in cellular process, MHC protein complex is first rank. Also, we see that the morphogen activity is top in the molecular function. And now we see that a pathway enrichment analysis from KEGG and reactome. In KEGG, allograft rejection is achieved first rank. And also we see that MECP2 regulates transcription factor RHSA is also a top rank in reactome analysis. Now, it's a very important result from protein-protein interaction analysis. In protein-protein interaction analysis, we use a server called network analyst server, and we also use a software called Cytoscape to rank the our hub genes. So we see the result of protein-protein interaction analysis through network analyst server. Here we see the our our result, and also we see the list of top 10 hub genes through Cytoscape's software. We see PPR gamma rank first, and the second rank is VDR, and ACTA1 is third rank. Also, another crucial part called co-regulatory connection between transcription factor genes and transcription factor microRNAs. Here we see that transcription factor hub gene interaction network. We see STAT3 transcription factors, CREB1 transcription factor, JE1 transcription factors, and the list go on. The important information is that this transcription factors, we use find out this transcription factor. We use the server called network analyst, and then we see the microRNA hub gene interaction map. We also use network analyst server to analyze this portion. We see that HSA MIR 144, HSA MIR 19A, and the list go on. So the last part is drug prediction. Last but not the least, that is the most crucial portion of our research. We see that 2 arachidonylglycerol rank first, and the second rank is phentin acetate, and the third rank is cannabidiol CTD. In this portion, we also use enricher server to find out the result, and I also use a database called DSIG-DB to find out our potential drugs or molecule to treat ALS. And then the last part, the conclusion part. So the study found DEGS and molecular biomarkers in ALS. These findings enhance comprehension of genetic foundation of the disease and the role of gene expression alteration in its course. The study highlights the vital role of early detection by the examination of both transcriptional and post-translational markers, potentially enhancing diagnostic and patient outcomes. And then this analysis not only identified significant DEGS but also emphasized potential interaction elements with the gene network, uncovering interesting pharmacological targets that may provide novel therapeutic strategies for ALS. Thank you, everybody. Thank you so much.